Hey, this is Malinki. Welcome to my channel, Voice of Malinki. Hope you all are safe and doing well in this pandemic situation. So today I will deliver the fourth lecture of COVID-19 lecture series, that is how to produce a vaccine. And if you are new in my channel, please don't forget to subscribe my channel. And if you like my video, please do like, comment and share my video. Okay, so the first step of vaccine production is to identify the antigen against which you need to produce the vaccine. And to identify the antigen, you require to have the knowledge of pathogen structure, route of entry, interaction with cellular receptors, subsequent replication sites, and disease causing mechanism. In case of COVID-19, the pathogen we know is SARS-CoV-2. Now we know its structure and genetic sequence. We know the route of entry, that is our nose and mouth. And then it targets one type of lung cell, that is type 2 pneumocyte. And the receptors are ACE2, which causes the interaction with the virus. The replication site is cytoplasm. In the cytoplasm of the cell, this virus gets replicated with the help of RDRB, that is RNA dependent RNA polymerase, and host cell ribosome. So I have already explained the disease causing mechanism of the virus in detail in my last lecture. So if you didn't go through them, please have a look once. I am providing the links in the description box. So the first step is complete. That is, now we know the pathogen life cycle. So the next step is immune control and escape. Now, from the first step, we know about the interaction between host and pathogen. Now we have to determine the immune response after the viral attack. That is, whether it is producing antibodies. So we need to determine different types of T cells. These are T cells. So we need to determine different types of T cells. We need to determine immune escape strategies during infection. Some viruses mutate very rapidly. This allows them to evade the antibodies generated by the infected cells. So this process is known as viral escape or immune escape. Now this is a problem to produce effective vaccines against some viruses. So we need to identify all of them. These informations will guide you to know the immune response of our body that will help us to proceed one step ahead in this vaccine development process. Because until and unless you know about the immune response and escape during a viral attack, it will not be possible to produce the vaccine. And now we know what happens in our immune system post-infection of coronavirus. So it is the second important step. So from the previous two steps, now we have identified the antigen. So antigen is selected against which we will formulate the vaccine now. Production of vaccine depends on some platforms, that is what type of vaccine it is. A vaccine should be of many types. So depending on the type, whether it is a viral vector vaccine or it is an inactivated vaccine or if it is an mRNA vaccine, the production methodology varies. Now, early vaccination were produced in vivo, like infecting calves with cowpox or rabbits with rabies virus, although most influenza virus vaccines are still made in chicken embryos. But these were more time consuming and stressful for the animals, right? So recently, vaccines are developed in the lab or in vitro. That is, we are culturing the animal cells in laboratory, and those are used for vaccine development. So those are faster compared to the in vivo and ethically cool. And mRNA vaccines, those are very recently produced vaccines, are representing a promising alternative to conventional vaccine approaches because of their high potency, capacity for rapid development, and potential for low-cost manufacture and safe administration. Now, preclinical testing is conducted with human cell culture, where scientists investigate how the vaccine impacts these cells using in vitro techniques. That is, if we treat our cells with vaccines, how do they react? How do they respond? Then they can survive or they die. So, if we get a great response in cell, then we use animal models, where a species of animal is selected and the effects of the vaccines are studied in vivo. It is also a fundamental stage for highlighting any potential risk relating to the use of vaccine. Side effects could be fever, skin irritation, muscle pain, discomfort at the injection site. So we need to identify the possible side effects. Here, we also evaluate the initial safe dose. That is, when we identify a vaccine, we actually don't know which dose will be most effective. So, the first thing is here to at least identify a safe dose. These doses may not be effective, but at least safe. That is, it wouldn't be lethal. Overall, the main aim of preclinical testing in vaccine development is to ensure that a vaccine is safe enough to be tested in humans. You cannot directly apply this vaccine in humans because you don't know whether this vaccine can cause some lethal effects. In the worst case scenario, death. So we are choosing some animal models in this preclinical targeting or preclinical testing. In this preclinical test, we also monitor whether these vaccines develop antibodies, and if antibodies are developed, how is its quality and quantity, whether it is able to combat the disease or not. Clinical trials are research studies performed in people before launching the vaccine in market. So now we come to people from cell culture and animals. Now clinical testing consists of 5 phases. Phase 0 to phase 4. Phase 0 is done with a very small number of people, usually fewer than 15. So, in the preclinical trial, we have determined the initial safe dose of the vaccine. But it was tested in animals, right? So, now we need to check whether these dose is also safe in human body. To do that, we just need very few people and we'll make sure that this much of dose is not harmful to humans. Well, till now we know the safe dose of the vaccine, but still we don't know the effective dose, that is, which dose will be effective enough to combat the disease. Phase 0 requires very short time, like not more than a month. If we are successful in stage 0, that is, now we know the safe dose in human, we will proceed toward phase 1. In this phase, we require small number of volunteers. 20 to 80 people are enough here. But these people must be healthy, who don't have any underlying health conditions. This phase aims to figure out the highest dose humans can take without serious side effects. These people will be very closely monitored to see how their bodies react to the medication during this phase. In this phase, we will also determine the best way to administer the vaccine, such as orally, intravenously, or topically. Here we will check whether these persons are experiencing any side effects or not. This phase takes 1 week to several months to generate a good amount of data. And if we are successful in this phase, that is, we identified the highest dose of the vaccine that didn't produce any lethal effect to human, then we move forward. So the next phase is phase 2. This phase involves several hundreds participants who are living with the condition that the vaccine is meant to treat. That means in case of COVID-19, we applied this vaccine in corona patients only. They are usually given the same dose that was found to be safe in the previous phase, that is phase 1. Now, participants should be monitored for several months or years to see how effective the vaccine is, and we need to gather more information about any side effects it might cause in these patients. When we are successful in phase 2, that is, we found the patients to recover and they didn't show any lethal side effects, we will move toward phase 3. Data we generated from phase 2 is actually not enough to demonstrate the overall safety of a vaccine. That's why we require phase 3, where we involve up to 3,000 participants who have the disease. So we need a large amount of data now. These phases can last for several years. Phase 3 trials are usually double-blind. That means neither the participant nor the investigator knows which participant is taking the vaccine. This helps to eliminate bias when interpreting results. The FDA or Food and Drug Administration usually requires a phase 3 clinical trial before approving a new vaccination. Okay. Now phase 4 clinical trials happen after the FDA has approved the vaccine. Now the vaccine is approved. So this phase involves thousands of participants and can last for many years. This phase is important to get more information about the long-term safety of the vaccine, its effectiveness, and any other benefits. So I think now you have some idea how a vaccine gets produced. So it's a huge matter, right? So can you imagine how scientists worked in the laboratory to make COVID vaccine so fast? It needs tremendous and extensive work. When we are at our home, busy in social networking, doing some politics over social media, most importantly, when we are safe under the roof of our home, scientists used to work day and night to generate vaccine for our well-being. If any issue occurs after the vaccination procedure, if anybody dies, you can imagine how society reacts. So, how much care a scientist needs to take care while going through this vaccine production? How much responsible they are? I was in a research field for more than 8 years, so I have some idea. I know how much struggle we do to invent a new thing. So, a small message to all of you. Please have patience. You will get your vaccine soon. Tons of vaccines are getting produced. We will all get them and will combat corona very soon. Let's work together. Let's not make fun of this. Be patient. And hats off to those scientists and volunteers who promise to save our life. And how exactly COVID vaccine is developed, how do they work in our body will be my next topic here. Please stay with me for the next exciting video. Bye-bye.